Okay, fine.
Yes,
Thank
we
you.
can hear you.
So we're going to pick it up from where you left it with Chavala. And as I mentioned earlier, my name is Lumwanga. That's my name. Lumwanga is my name. So we're going to continue with the introductory part to medicinal chemistry. So based on what, I don't know if we can remember anything that we looked at last time. And that medicinal chemistry is Chavala. As far as the introduction was concerned. Anything, it can be a key term or anything that you can remember. Okay, so today we're going to introduce medicinal chemistry to you guys as nothing but advanced pharmacology. I'll describe that as being advanced pharmacology because under pharmacology, our main interest is the interaction that exists between drugs and biological systems. That is what we get to look at. Sorry for that interaction. So you get to look at the interaction between drugs and biological systems. And you get to describe a drug as being any compound that is of known structure, that isn't a vitamin, that when administered into the organism has the capacity of bringing about a biological effect.
You can get your feedback.
So medicinal chemistry is the study of this year, but just at the deeper level. So it is nothing but advanced pharmacology. And in pharmacology, what you get to look at are these drugs and the biological effect that they get to have in our bodies or any live systems. But according to what has been written here, medicinal chemistry is, yes, the study of chemistry. Don't forget about the chemistry, at least up to this point. Focus on the medicine and what's the interaction that exists between this medicine or drugs and biological processes. But it goes a little bit deeper than pharmacology because you're not only looking at the effect that these drugs, which have been colloquially termed medicine, although there's a very big difference between medicine and drugs. So the relationship between drugs and biological systems. But you go further by looking at the chemistry of these drugs. What is it that they're made out of? Why is drug A compared to drug B? Or why is drug B superior to drug B? What's the difference between propranolol and atenolol? What's the difference between diclofenac and aspirin? What's the difference between drug A and drug B? So at the core of medicinal chemistry is nothing but pharmacology. It's nothing but pharmacology. But this pharmacology is deep in that you are looking at quite the right drugs, but you're looking at the chemistry of drugs. It is also deeper in the sense that you also get to look at this other aspect of drugs. Under pharmacology, you get to discover existing drugs. That is what you get to study. You get to study existing drugs that are already on the market. But armed with the knowledge of medicinal chemistry, you should be able to design and develop new drugs. So by definition, you should appreciate that there's a very strong connection between pharmacology and medicinal chemistry because you get to study pharmacology as you're studying medicinal chemistry, but you get to delve a little bit deeper in the sense that you have to know the chemistry of those drugs. What is it that they're made up of? What functional groups do you find in those molecules? How do they get to interact with the receptors? Why is this drug able to cross the blood-brain barrier unlike this other drug? Why is this drug able to be given orally unlike this other drug when they have the same parent compound. That's basically what you get to look at under medicinal chemistry. So looking at the bridge or the integration between chemistry and drugs or medicine by designing and developing drugs based on their structure activity relationships, and understanding the biological processes. But to make things simple, you can just describe medicinal chemistry as being just advanced pharmacology, because it gets to feature one, chemistry, two, in addition to studying already existing drugs, after understanding this course, you should be able to design and develop new drugs. So there's a component of drug discovery, drug designing and development. Then there's this other component here. Actually, this is a tool that helps you discover, design, and develop drugs, the structure activity relationship, and we're going to look at that in a bit. So that's what medicinal chemistry is all about. So firstly, we are going to look at the chemistry of drugs that are already in circulation. We're also going to look at why they behave the way that they behave at molecular level, meaning that we have to draw their structures, look at them, find out why they behave the way that they behave, find out why certain drugs have better activity compared to other drugs, find out the relationship amongst a certain class of drugs. I'll give you an example of penicillins. Penicillins come in very, very huge classes. No, they're just 4 classes of penicillins to be specific. But they come in a wide array of characteristics. So we have penicillin G. I'll give penicillin G as an example. Penicillin G is the prototype of all these penicillins that we know: amoxicillin, cloxacillin, what else can we think of? Amoxicillin, cloxacillin, ampicillin, and the friends. But you're going to notice that amoxicillin is quite different from cloxacillin, and cloxacillin is completely different from ampicillin, and ampicillin is completely different from penicillin. So we're going to look at how all these drugs, first of all, were designed and how they were developed, the chemistry that was behind the design of these drugs, and how they get to interact with biological processes. And our study of the structures of these drugs is going to be aided by this here, the structure activity relationship, and that's what we're going to look at in a bit. So some of the main components that we are going to look at under this course, and the ones that you're going to be run through as long as you remain at UNZA 4th year, and you'll be there, of course, this whole year. The first thing that you're going to look at is the drug discovery and development process. This is actually a whole component which is going to be taken, by Mr. Mutati. So Mr. Mutati most likely is the one that is going to be taking this whole component. So under this component, what you're going to appreciate is the effort that it takes to discover a drug and actually put it out there on the market. A long time ago, there was no penicillin. A long time ago, there was no amoxicillin. A long time ago, there was no diclofenac. A long time ago, there was no warfarin. A long time ago, there was no insulin. At some point, all those drugs were nothing but ideas that either someone stumbled upon or someone thought they could exist. So at some point, those drugs were not existing until someone had to discover them. And after discovering them, they had to be developed. And after some consecutive steps, which are quite expensive and time consuming, that drug which was discovered was later approved to be used by the general public, and there we have it on the market. So this is a very, very long process that you're going to be looking at. How is it that drugs get to find themselves on the market? Maybe just to give you an idea as to what is involved. I think the first step that is involved in drug discovery is, yes, this has been mentioned. This involves identifying therapeutic agents. And the first step is this one here, finding lead compounds. A lead compound is just let me not even call it a drug, I'll call it a molecule. It's a molecule that has some desirable pharmacologic effects. It's any molecule that has some desirable pharmacologic effect. And this is the one which is usually discovered first in this whole drug discovery and development process. The first compound that someone is going to encounter is going to be termed a lead compound. It's not going to be useful in the general public, neither can it be tested in human beings, because those compounds, upon being discovered, have a lot of downsides to them. They can be very, very toxic. Despite them having desirable pharmacologic activity, say someone discovers a drug which has the ability to inhibit the replication of cancerous cells. That first molecule someone is going to encounter is going to be termed a lead compound. And that ability of that drug to inhibit the replication of cancerous cells is going to be a desirable pharmacologic effect. But the problem is, it could be that that molecule that someone encounters first could be toxic to the heart, it could be toxic to the liver, or it could be that that drug has a very low half-life. It could be that that drug has a very low half-life, say of 2 seconds or 2 minutes. If such a drug has such a very low half-life, that means you have to be administering that drug every, I don't know how many hours or seconds. If it has such a very low half-life, that means that the dosing frequency should be very high. Is that desirable? No. That's very, very undesirable. It could also be the case that that drug is not available for oral administration, probably because it's degraded by the acid which is found in the stomach. So it could be unavailable, for oral administration. But then if you had done a drug which is going to be used to stop the replication of those cancerous cells, you wouldn't manage having someone injected this drug that you're first going to discover, actually this lead compound, forever and ever up until they stop, up until they, okay, some people get cured of cancer. Yes. But you wouldn't intend for someone to keep on injecting themselves, or for them to be in the hospital, they can be given this IV drug, because it can be given orally for the rest of their life up until the cancer gets to disappear. So the basic idea is the first molecule that scientists get to stumble upon isn't a perfect drug that can even be used in human beings or even animals. They're very toxic, or maybe they may have poor pharmacokinetic properties that may make them not possible to either even be tested in human beings or even animals, or even be used in a clinical trial. So that's basically that. Then if you discover a drug, of course, you have to optimize some of the properties. Okay, yeah, fine, we have this drug. No, not really a drug. We have this lead compound. What we know, it has this desirable pharmacologic property of stopping the replication of cancerous cells. But then it's toxic to the liver. Its half-life is very low. It's not available for oral administration. What are we going to do? Okay, let us find out the reason why this drug is toxic. Studies can then be done. If certain groups are identified which are making the drug toxic, they can be gotten rid of. If the reason why that drug has a very low half-life can be determined, some structural modifications or optimization strategies can be put in place. So those properties which are desirable can be fostered, whilst the bad properties can be gotten rid of. Then from there, you have to test this drug in human beings. No, firstly, you have to test that drug on cultured cells in petri dishes, then test that drug in rodents and dogs, then test whether they have a pharmacologic effect. You have to test as to whether they are toxic. You have to test as to whether they are carcinogenic or they can bring about cancer. You have to test as to whether they toxic to the fetus. So meaning you have to get at this stage, that is, if you're using rodents and sometimes dogs, you have to get a pregnant rodent, inject that drug which you're trying to optimize, inject that drug into a pregnant rodent and see whether it has any adverse effect on the fetus. And that takes time. And after that, you have to test that drug in human beings. After testing it in human beings, then you can find them itself on the market. So this whole process of drug discovery and development, which involves discovering a lead compound and getting rid of all undesirable properties that it may have, whilst promoting the good properties that it may have, and testing it with a goal to it being on the market is what drug discovery and development is all about. This is a whole component that you're going to look at with me sometimes. I don't know if you have any questions up to this point. Hello?
Queremos Andrea.
Do we have any
I
questions?
think it's clear.
It's clear. Good. So one of the components that you're going to look at under medicinal chemistry is how drugs get to find themselves on the market after discovery. The good part is some drugs are just stumbled upon, meaning people just discover them by accident. For example, penicillins, whilst others are discovered by means of metabolism studies. Say you have this individual here. This individual is given drug A, and this individual, the same individual is also given drug B. Then after this person is given this drug A and drug B, and okay, let us forget about drug B initially. So this person is given drug A, and then after some time, the blood from this person is extracted, some blood sample is taken, and then some metabolites of drug A is noticed. It could be that this drug A, metabolite of drug A, could be pharmacologically active. Remember, under the metabolism of drugs, metabolism doesn't always entail that the drug is going to lose its pharmacologic activity. Sometimes metabolism, either by means of phase 1 or phase 2, can also result into the retainment or even increase in the activity of the drug. A good example is codeine. Codeine is administered as a prodrug. It needs to be converted to morphine for it to work. So the same applies to certain drugs. Sometimes some other compounds are given, some other compounds could be given, and by studying some metabolites that are produced in the body and their pharmacologic activities, some drugs have been discovered in that way. A good example is paracetamol. Paracetamol was discovered in that manner. There was a certain compound, I've forgotten what that compound was, but it was administered in people under a clinical trial. Then a certain compound was noted to have some analgesic effects. Analgesic meaning stopping pain, some analgesic effects, as well as antipyretic effects. So basically that's it. There's a lot that is in store for you guys. But basically the idea is how are drugs discovered? What are those methods that are used to discover drugs? And how do they find themselves on the market? Then the next thing that you are going to look at is this, a very big topic also. We're also going to look at drug targets. Remember, what did we say pharmacology is all about? Pharmacology is a study of drugs. But then, what is a drug? A drug is any chemical compound that is of known structure and has the ability of inducing a biological effect. In a live organism, that is. So for that drug to produce these biological effects, it has to, at least in most cases, a drug has to interact with a receptor or a target. A drug has to interact with a receptor, a target, for it to bring about that biological effect. So the different targets that a drug can interact with, some of them could be membrane embedded receptors. This is one of them. So membrane, under membrane embedded receptors, you have some ion channels, you have some, so you have some ligand-gated ion channels, you have some voltage-gated ion channels, you have some enzyme-controlled receptors, and you also have some guanyl protein coupled receptors. So that drug has to interact with these membrane embedded receptors or receptors that are found in the membrane, which could either be ligand-gated or voltage-gated ion channels, enzyme-linked receptors, or GPCRs. At other times, the drug can interact with intracellular receptors. Intracellular receptors, these could be some enzymes that are found inside. Some enzymes that are found inside the cell. Sometimes these enzymes will be found outside the cell. Maybe let's give examples of ligand-gated ion channels. So examples of ligand-gated ion channels that drugs can interact with, for example, may include anyway, yeah, the nicotinic receptors. Nicotinic receptors, especially the nicotinic muscular, those found on skeletal muscles. These are found on, yes, they're found on skeletal muscles. And if acetylcholine can bind to those nicotinic muscular receptors, they can open, leading to the influx of sodium. So this is just an example of ligand-gated ion channels. Then there are some voltage-gated ion channels, enzyme-linked coupled receptors. These may include insulin receptors, GPCRs. These are very common. The beta receptors, and there are quite so many GPCRs. Then you have receptors that are enzymes in nature. There are so many enzymes that can act as drug targets. Can you think of enzymes that are used as receptors or drug targets in the body? There are so many. Acetylcholinesterase acetylcholinesterase found at the synaptic cleft in the CNS and the peripheral nervous system, good targets of anticholinergic agents, for example, hydrophonium. So basically, yeah, carbamates as well as organophosphates. But there's so many enzymes that are inhibited. Others are, yeah, you also have monoamine oxidase, MAO responsible for the degradation of endogenous catecholamines. So you have MAO-A and MAO-B inhibited by drugs such as selegiline and the friends ABCD. So yeah, you obviously have to talk about drug targets too. If you're going to look at pharmacology in details, you're going to look at what the drug looks like and how it's going to be interacting with that particular receptor. So it's only logical that if you're going to look at this drug at so much of a deeper level as to figuring out what atoms make up this compound, it's only logical that you look at this target also at a deeper level so that you can look at really what is going on between this drug here, whose chemistry you have to study at depth, and the active side of this drug target. It could be an enzyme, it could be a receptor like you mentioned. Sometimes it could be a segment of DNA. It could be a segment of DNA, as in the case of anti-cancer agents. So basically that you have to look at these molecules in the body that are involved in disease process. Drugs interact with these targets to modify their function and produce a therapeutic effect. So yeah, apart from learning about how drugs get to find themselves in the market, you have to look at what molecules the drugs are interacting with, because you're going to study these drugs at a very, very deep level. So it's only logical that you get to look at what they are interacting with to produce that biological effect, also at a very deep level. But is it of any use if we can have a drug that can perfectly interact with this receptor, but it can't reach the receptor? It's of no use. If you had a cure for, say, cancer, but then no matter what you do, that drug cannot be absorbed or it can't leave blood vessels. Say it can be absorbed. That means if you take it orally, it's going to pass through the GIT and it's going to be expelled. If it can't leave the bloodstream, that means, say, if this person has cancer or whatever it is, if we have cancerous cells here and this drug that you've discovered, quite right, it's a very good drug and it can stop these cancerous cells from multiplying. But of what use is it if this drug can't reach the cancerous growth? So this principle comes in. So after knowing drug targets and the structures of drugs and how they come about, how they are put on the market, the process that is involved, you also need to know the pharmacokinetics and the pharmacodynamic properties of that particular drug. So pharmacokinetics is, as you looked at, it's nothing but this here, the ADME process. Yes, you have a drug which can, for, okay, yes, for arguments, not really for arguments, let us just make a wild guess. Let us say you've discovered a drug which can cure HIV. But if it can't get absorbed, if it has pure distribution, if it has, say, super fast metabolism that it doesn't even last a second in your body, or if the excretion of that drug in the body is too high, so much that a person needs to be taking that drug every after a minute, it wouldn't be of any use. So actually, this takes you back again to this here. This takes you back to the chemistry. So as you are studying the chemistry of drugs that are already existing and drugs that you're going to discover, based on their structures, the structure is going to give you a lot of information about what the half-life of the drug is likely to be, what the absorption of the drug is likely to be. So what the absorption is likely to be, what the excretion of the drug is going to be, what the half-life of the drug is likely to be, what the protein binding capacity of that drug is likely to be. So all that information gets to come from the chemistry. of these drugs, which you study, which you get to study under which course, which you get to study under medicinal chemistry. So medicinal chemistry gets to help you understand a lot of things. In addition to understanding pharmacokinetics, you get to appreciate the pharmacodynamics. Pharmacodynamics is the effect that drugs have on the body. So it takes you back again to what? It takes you back to this year. Because for drugs to exert a pharmacologic effect or a physiologic response, they have to interact with some receptor. So this is what medicinal chemistry is all about. One, drug discovery. Two, looking at drug targets and how drugs gets to interact with those targets, what bonds are involved, what uh, type of interactions are involved between the drug and the active site of that particular drug targets. If it were an enzyme, do we have any hydrophobic interactions between the drug and the targets? Do we have any ionic interactions between the targets or not between the drug? The drug needs to have good pharmacokinetic properties if it's going to be useful, which is actually something which is worked on during this process that I mentioned earlier here. So once you get to discover these lead compounds, it may not have, okay, all its properties may not be appealing or good at the time, but you have to find ways in which you can optimize them. You have to find ways in which you can get rid of the bad properties and find ways in which you can boost those good properties of the drug. For example, potency, the activity that it has, and the specificity of that drug when it comes to binding to the receptor. One thing that we didn't mention that was in the definition of what medicinal chemistry is this component here. What is actually going to help us to design and develop drugs, especially new drugs, develop new drugs, is studying their structure activity relationship, the SAR. What the SAR is this? The SAR is us trying to find out this here. It's us trying to explore the connection between the chemical structure of the drug and the biological activity of the drug. So here you have to come up with the structure of the drug. I'll give an example of say, penicillins. So let me write the SAR of penicillins here. So let me draw a structure of the general structure of penicillins. So penicillins have in that structure. Numbered from 1 carbon. Oh yeah, that is there, the sulfur there is position number 1, that's position 2, that is our position 3, that is our position 4, this is our position 5, this is our position 6, this is our position 7. So the structure activity relationship is the connection between the chemical structure and the functional groups present on the structure. So looking at the chemical structure and the functional groups present, looking at the relationship between the structure of the drug and the functional groups present and the biological activity. The structure activity relationship is you looking at the structure and saying, so what's the relevance of this alpha here? Is it really relevant? What if we remove this alpha and replace this with carbon? What would happen? Or what's the relevance of this carboxyl group that we have there at position 3? Is it necessary? What if we added an aldehyde here? Would it still work? What's the relevance of this carbonyl that we have there? What if this ring here were slightly bigger? Would it have any bearing on the activity of this drug? By the way, this is a penicillin. This is a general structure of a penicillin. So the structure activity relationship is you making that connection between the different groups present on the structure and the biological activity. This biological activity could mean the pharmacokinetics of the drug, it could mean the pharmacodynamics of the drug, it could mean the interaction of the drug with the receptor, interaction with receptor. It could mean so many things. So it's you saying, what's the relevance of this? What's the relevance of this? Okay, I'm looking at these hydrogens which are position 6 and 7. They seem to be in cis relationship because they're both, yes, they're both going into the page because of these dotted lines. What would happen if one of these were like that? Would it have any bearing on the structure? That's what the structure activity relationship is all about. And once you know what this does, what this does, what this does, what this does, what that does, what that does, what that does, and the relevance of all these groups, then you can start playing around with different functional groups. Say if you discover that this is responsible for making the drug, this is responsible for absorption, and you discover that this drug has poor absorption, you're going to find out why this functional group here leads to poor absorption. This is just a supposition. We're supposing this. Why does this group lead to poor absorption? Okay, fine. After you find out the chemistry behind why this is making this whole penicillin have poor absorption, then you're going to figure out, using chemistry, of course, which other groups could substitute this here. So that if you replace them, if you replace this carboxyl group with this other group, say group B, it would improve the biological activity, not the absorption, because that's what we're looking at. So basically, that's what the structure activity relationship is about. You're looking at the structure of the drug, which has to be drawn, it has to be in full, and you making that connection between the different structures that this drug has and the relationship. And actually, after doing the structure activity relationships of drugs, let us even just make some. Let us try to come up with a structure activity relationship of penicillins as an example. So this is just an example. We're going to look at this later. So if I were told to come up with a structure activity relationship of penicillins, which you guys are going to look at, we are going to look at, after coming up with different types of penicillins, it has been discovered that the first sulfur, the sulfur here at position 1, is not that necessary. So this is not necessary. It can be substituted with, say, carbon, and that would be fine. This is what analogs of this penicillin, which the S has been substituted with C, have shown. Whether they have a C or whether they have an S, there's absolutely no problem. Then with respect to position 3, it has been discovered that the position 3 has to be ionized in the body or at the receptor site for it to interact with the target. It has to exist as C or O minus. That's one thing that has been discovered and proven. And it has also been discovered that this carbonyl here at 5, position 5, is very important. So the 5 carbonyl is important. What does it do? It's responsible for the mechanism of action of penicillins. So this here is necessary. So yeah, basically after doing all of that, coming up with the structure activity relationship, seeing what this does, what that does, what that does, what that does, which is of course given in various books and you're going to be learning such, you will discover that there are certain groups which are very necessary you discover that there are certain groups which are necessary. There are certain groups which are necessary. And then there are certain groups that can be modified. They are not that necessary. You can even modify them. So after studying the structures of these drugs, there are some certain parts of these chemicals, of these drugs, which are very, very necessary. Any modifications to these structures result into the loss of these drugs. They lose activity, they won't do anything. If at all you want to discover a new drug and you tamper with that part of that molecule, which is very necessary for it to do the work that it does, and you modify it, then you discover that the drug that you're going to come up with is not going to have that pharmacologic effect of the parent compound which you're trying to model after. So yeah, there are certain parts which are necessary. Let me show you this in the next slide. So yeah, basically, yeah, those components which are important are known as pharmacophores. So the pharmacophore is the essential arrangement of functional groups in a drug molecule which is responsible for the biological activity. Any alterations to these groups minimizes or reduces completely any pharmacologic activity. Any pharmacologic activity possessed by a drug. It doesn't represent the entire structure, but highlights the key features responsible, but highlights the key features essential for interaction with the targets. Let me use this as an example here. So we have various penicillins here. These are all penicillins here. These are their structures. But so yeah, this is ampicillin, not that common nowadays. Amoxicillin is very common. Cloxacillin is very common. Also, dicloxacillin is not that common in developing countries. Nafacillin, yeah, but not that common too. Oxacillin, not that common too. Penicillin G, very common. Pen V, very common too. What you're going to notice is observe this structure that I'm going to encircle. So look at that structure. Look at this structure also. Look at this structure also. Look at this structure also. Look at that structure also. Look at that structure also. Look at this structure also. Look at this structure also. You won't spot any difference in these structures that have sequence. All these structures have this structure. So the whole thing that I've circled here is found here. It's actually the same thing that I've circled there. This carbonyl, that hydrogen, that square there. Then you have that pentagon looking like ring. You have S there. Then you have this carboxyl group. Then you have these three methyl groups. That's basically what you have. Those two there, carboxyl, the square, NH. That's what you have there. 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 So all these that you are seeing there, all these that you are seeing there, all these that you are seeing there are very, very important for pharmacological activity, and they are called the pharmacophore. The only difference that you can see is this part which is attached, that 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 part which is attached. So if you are to compare, say, penicillin G to amoxicillin, penicillin G actually is not even available for oral administration. Penicillin G is available only in IM or IV. Is available only in IV or IM administration. Because of these differences between this group that you are seeing here, this benzyl group, and this benzyl group here, and this, which group? Yeah, this here is amoxyl. So because of the differences between, because of this amino group that you are seeing here and the hydroxyl group, which this penicillin G does not have, these two Actually, this one here makes it possible for amoxicillin to be given orally. The chemistry behind that is something that you're going to look at. The chemistry behind that is something that you're going to look at. But I just want you to appreciate the fact that there are certain functional groups which should not be tampered with. They should always be present. And those are needed for the pharmacologic activity of the drug. If a drug does not have those structures, then if the drug didn't have those structures, then we wouldn't have had those drugs with that activity of inhibiting the cell growth, particularly the cell wall synthesis in penicillins. We are almost winding up anyway, hopefully. So that's basically what that is. So you have some certain parts of the molecule which are needed for pharmacologic activity. Then you have those which are not that necessary for pharmacologic activity. Okay, good. So there are those that do not directly, I can see text which is up there. So there are those that do not directly participate in the binding, that do not participate in the direct binding, but they are there to influence the pharmacokinetic and the pharmacodynamic properties of the drug. These can be modified and the structure will not lose, the drug is not going to lose its pharmacologic activity. And that is what we have there. This is what we have, just a second. That is what we have in this slide here. So all these structures here, all these are oxophores because they can be modified. They get to differentiate the pharmacokinetic properties as well as pharmacodynamic properties of this drug here, penicillin, to this drug here, and amoxicillin. These are oxophores, but they all need to have the pharmacophore. The difference is the oxophore. The oxophore can be modified, and that's fine. The drug won't lose its pharmacologic activity, but if you change the oxophore, you get to observe some changes in some pharmacokinetics as well as pharmacodynamic properties. It could have better absorption, or it could have a longer half-life, or maybe it can become resistant to the acid which is in the stomach. So basically this is what you get to change. This is what you get. So in case you want to discover any penicillin in the future, this is what you should modify. Modify that, don't tamper with this. This is the pharmacophore. So basically that's that. So that's your pharmacophore, and that's your oxophore, the part which can be modified. And there are certain groups that can be, anyway, this can be explained in the next slide. So now, depending on what is used as the oxophore, remember, is the part which can be modified. And if these modifications are done, the pharmacologic activity of the drug is not lost. Only the pharmacokinetics of the drugs, as well as the pharmacodynamic properties of the drug, as well as some other properties are affected. Other properties. But if, say, drug A has the ability of, stopping seizures, and it has a certain pharmacophore, and we come up with drug B, which has the same pharmacophore as drug B, as drug A, but we just get to change the oxophore. That means we're going to come up with a drug which is going to be doing the same thing as drug A as far as the pharmacologic activity is concerned. It's going to be stopping seizures, but then it's going to have slightly different pharmacodynamic properties or pharmacokinetic properties. It can have a better absorption or probably poor absorption, or it can start to last longer in the body, or maybe it can cross the blood-brain barrier. That's the interesting part of what do you guys again? Medicinal chemistry. But then the effect that modifications to the oxophore has So yeah, we said we have the pharmacophore. This shouldn't be tampered with. It's needed for pharmacological activity. Then you can also have functional groups, other functional groups, which we're terming the oxophore. So now the certain groups of the pharmacophore can be changed. Yes, the pharmacophore can be changed. So some certain groups can be added to it. Then those groups that can be added to the pharmacophore can either be hydrophobic or they can be hydrophilic. They can be hydrophobic or hydrophilic. Now, these modifications and the effect that they have on the whole drug have been summarized for us in this table here. They have been summarized for us in that table. So should it be the case that we have, say, drug A has this pharmacophore, and drug A has this pharmacophore, and this oxophore, and we come up with drug B, which has the same pharmacophore as drug A, but it has a slightly different oxophore. It has a slightly different oxophore, which is in this case now more hydrophobic. We're supposing that this is a hydrophobic pharmacophore. We had this group here, but we replaced this group with something which is much more hydrophobic. What is going to happen is the overall effect of this change, the overall effect that this change is going to have on this drug is that is going to improve absorption, that is also going to increase protein binding capacity, that is also going to increase the distribution patterns of the drug, that is also going to make the drug last longer in the body by making those modifications. So if, say, you have a drug and you want to, say you have this drug, you discover it, but then you notice that it has very poor absorption. So what do you do? First of all, figure out what the SR is. Draw the structure of that drug. After drawing the structure, what do those groups that are present on the drug do? What does this carboxy which you have at this point do? Does this nitrogen that you have at this position do? What's the relevance of this? Can this be modified? After finding out what the pharmacophore is, then you know what you don't have to tamper with. Then, say you suppose this drug has this pharmacophore that you discovered after your ACR, you discovered this segment of the drug is necessary for pharmacological activity. And you discover this part can be tampered with, and this drug has a very low half-life. But you like this drug. It is a very, very important drug because it has a certain pharmacological activity. You can make this oxophore more hydrophobic. If you make it more hydrophobic, that means the absorption of the drug is going to be improved. The protein binding is going to be improved, meaning it's going to last more in the body. If its protein binding capacity is very high, that means it being excreted and ultra high filtration in the glomerulus of the nephron is going to be low because it's bound to proteins, and proteins do not get to pass the slits that are found in the glomerulus. Then the distribution is also going to be high because it's hydrophobic. If it's hydrophobic, then it can easily cross towards the phospholipid bilayer. So that means the distribution of the drug is going to be very high. It can be found in virtually every tissue of the body, including the intracellular fluid. That means its half-life is also going to be high because most of the drug is inside the cells, it's inside the interstitial fluid, it's not inside the blood. That means the filtration of this drug in, say, the kidneys is going to be very low, and its metabolism in the liver is also going to be very minimal. So basically that's it. The opposite happens if you add a hydrophobic group, or a hydrophilic group rather. If you had a hydrophilic group, say NH2, if you had some groups that are just hydrophilic, you had NH2, if you had the carboxyl group, those groups make it hydrophobic, make the whole drug hydrophilic rather. And if it's hydrophilic, the drug is going to have poor absorption, it's going to have poor protein binding capacity, poor distribution, and it's going to have a very low half-life. So basically, that's the introduction to medicinal chemistry. You keep on coming back to this. You keep on coming back to this. I don't know if we have any questions, but that's what we thought we should look at. So a brief recap of what we've looked at. We look at medicinal chemistry as being nothing but advanced pharmacology, because you not only get to discover the activity that drugs have on the biological process, but you get to look at their chemistry, you look at their structures and the functional groups present and what they do. Then using that information, you get to discover, design, and develop new drugs. Armed with that information, you're going to look at this also. You're going to look at how drugs are discovered and developed and how they get to find themselves on the market. Then if you're going to discuss and study the effect that drugs have on biological systems, then you have to look at what those drugs get to interact with, the various receptors and what is going on. So actually in the medicinal chemistry, you'll be looking at the structure of the receptor and what is going on. If you say beta blockers get to occupy the beta receptors, the beta 1 receptors that are found on the heart, what really are you talking about? What bonds are involved between that beta blocker that you're talking about and the beta 1 receptor itself? What does the beta 1 receptor look like? What does that beta blocker cyclopranol look like? What is really happening here at molecular level? You have to look at the pharmacokinetics too. Then you need to know the different functional groups present on those drugs and their relevance. What does that do? 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 And that's what the structure-activity relationship is all about. Once you know what each does, then you can find ways in which either you can promote that thing or you can get rid of that group if, say, it's responsible for the toxicity of that drug. So these are notes for you to look at if you get to have some time. So the important parts of that drug, after the SR has been drawn, those parts which are responsible for pharmacological activity, that part is known as the pharmacophore. Then the other part, which can be modified, is known as the oxophore. The oxophore gets to govern the pharmacokinetics and the pharmacodynamic properties of the drug. And we looked at various changes on the oxophore and the effect that they can have on the drug as a whole. I don't know if we have any